Welcome back as we continue to work our way through Chapter 2. This is a continuation of Section 0.4, and this video is about the macromolecule lipids. At the end of this video, we should be able to understand the structure and function of lipids. Lipids are, I believe, the most diverse macromolecule. They have the most variety in how they look and jobs that they do. And it can get a bit complicated, so we're going to go nice and slow, step by step. And as always, if you have any questions after watching this video, please send me an email and come to office hours, and we can talk through anything that you're struggling with. So a characteristic of lipids in general is they're hydrophobic. Okay, hydro, water, phobic, fearing. So these are things that don't mix with water. They're organic molecules, meaning they have lots of carbons. Their backbone is made of carbon. And they also have a lot of hydrogens compared to the number of oxygens that they have. If I eat 1 gram of carbohydrates, I get 4 calories of energy from it. If I eat 1 gram of lipids, I get 9 calories of energy. So lipids have a much higher calorie content per gram than carbohydrates or proteins. And we have 5 types of lipids that we're going to be talking through in that order today. So let's start with fatty acids. Fatty acids are just a chain of carbons and hydrogens. Okay, they'll usually have a carboxyl group. Here's your carboxyl group. C, double bond to an O, and then hydroxyl on the other side. That's our carboxyl group. And then the methyl group, CH3, at the other side. We have 2 varieties, saturated and unsaturated. So let's think about the word saturated, because this is going to help you logic your way through what is saturated and unsaturated, say on an exam, say next week. So saturated means it's full. If I have a towel that I'm using to clean up some spilled water, the towel is saturated when it cannot hold any more water. My fatty acid is saturated when it has the maximum number of hydrogens and cannot hold any more hydrogens. Okay, so this is a saturated fatty acid. Every carbon has 2 hydrogens all the way down this chain until we get to the methyl group. The other is unsaturated fatty acids. And unsaturated fatty acids do not have the maximum number of carbons. That's because, they do not have the maximum number of hydrogens. That's because they have a double bond somewhere in the tail. And that double bond means they're missing 2 hydrogens. So it's not saturated. It is not full of hydrogens. If we somehow broke this double bond, we could add in 2 more hydrogens to the structure. What this means is our geometry here is bent. This unsaturated fatty acid tail has a bent shape. So imagine, let's go back to our saturated for a second. If I have a whole bunch of these saturated fatty acid tails and they're all packed together, they're going to pack together or stack kind of like bricks, nice and neatly, and they're going to be able to get real close together, and they're going to be a solid at room temperature. Think of butter, think of pork fat, or fat on the edge of a steak. Those are saturated fats. They're a solid at room temperature. Things that are unsaturated, think of corn oil or peanut oil. They are liquid at room temperature because the tail is bent, and that geometry makes it much more difficult for them to stack together nice and neat. The bent tail keeps the molecules kind of in a tangle, and the macroscopic results of that is being a liquid at room temperature. So next we're going to talk about a triglyceride. A triglyceride is the storage molecule, the way we store fats in our bodies. And if we are eating animal products the way we would consume fats in those animal products. So a triglyceride is made of 3 fatty acid tails. Okay, just like we looked at before, there's one fatty acid tail, second fatty acid tail, and a third fatty acid tail right there. And they're bound all together to glycerol. Okay, so we got the glycerol up here. When we have liquids, we call them oils. When we have solids, we call them fats. They can be saturated or unsaturated. And we're using these as energy storage. If you have anywhere on your body that is a little chubby, and if you poke at it, it jiggles, that is your adipose tissue. Think of your tummy or maybe your underarms. That's your adipose tissue. That is stored energy. It keeps you warm, it absorbs shock, and it also will give you energy if you're ever in a situation where food is not available to consume. So, this is a dehydration synthesis reaction. Okay, we've seen this before, but let's look at it again. So, I have my first fatty acid tail, and we can see right quick we have the maximum number of hydrogens, so this one is saturated. We have a second fatty acid tail. This one is a bit longer, also saturated. And here we have a unsaturated fatty acid tail. This one has 2 double bonds and therefore not the maximum number of hydrogens. And if we were to draw this a little bit more correctly, it would have 2 bends in the tail. And then we have the glycerol up here with 3 hydroxyl groups. And we're going to be concerned with the hydroxyl groups, 1 on each of the fatty acid tails. We're going to remove 3 waters. So we'll remove hydrogen, oxygen, hydrogen, hydrogen, oxygen, hydrogen, hydrogen, oxygen, hydrogen. And when we link the 3 fatty acid tails to the glycerol, we're going to end up with 3 waters plus a triglyceride. We have 2 different forms, trans fatty acids and cis fatty acids, and it has to do with the geometry around any double bonds that we have. Trans fatty acids take on a really strange geometry. They're a shape that is really difficult for our enzymes to break down, so they stay circulating in the blood for longer, and they're more likely to stick to your arteries, and therefore we have an increased risk of heart disease if we eat trans fatty acids. Trans fatty acids are produced when cooking oil is overused and it's heated at high temperatures for a very, very long time. Think of if you had a fryer at McDonald's for the French fries and the oil wasn't changed as often as it should, and the integrity of the oil started to break down, and some of that damaged oil is on the fries when you eat them. That's when you would consume trans fats. Cis fatty acids have more typical geometry around the double bonds, and they don't carry the increased risk of heart disease that trans fatty acids do. Here's a quick depiction of a trans fatty acid and a cis fatty acid. Our next type of lipid is phospholipids. Phospholipids make up the cell membrane. They are critically important in the integrity of all of your cells. Without them, we would be in some serious trouble. That is for sure. So let's take a look at these. What's unique about phospholipids is they're amphipathic. That means they have a portion that's hydrophobic and then a portion that's hydrophilic. Very few things have this kind of structure. Okay, so your fatty acid tails are hydrophobic, and these are the same tails that we saw in fatty acids and triglycerides. Okay, so we've got our hydrophobic long tails made of carbons and hydrogens. They could be saturated, they could be unsaturated. And then we have a phosphate group up here at the head, and that is hydrophilic. So this portion is very happy to interact with water, and the tail portion really does not want to interact with water. And how these guys arrange is in the cell membrane. If we have aqueous solution or a water-based solution outside of the cell, and also aqueous solution, water-based solution on the inside of the cell, the phospholipids will line up. So the head of the phospholipid is facing the aqueous solution on the inside, and then we'll have a second row lined up. So the phosphate head is facing the aqueous solution on the outside, and the fatty acid tails will stay in the middle. And this makes a selectively permeable membrane. Some things can go through pretty easily. Some things can't go through at all. And this helps us to regulate what goes into our cells and what comes out of our cells and keep that in balance. Eicosanoids. These are things that are similar to hormones. Some things that they play a role in are inflammation, blood clotting, the contractions that are involved with labor. Specifically, the eicosanoid that's important there is called a prostaglandin. And our last one is steroids. Cholesterol is the steroid that all the other steroid lipids come from. So we get a lot of negative perception of cholesterol. It's actually super important for us. Your brain has a ton of cholesterol in it. If you were to wipe out all the cholesterol in your body, your brain would stop working. Also, we have cholesterol kind of mixed into the phospholipids that make up our cell membranes, and it gives strength and integrity to those cell membranes so they're not so wishy-washy, flexible, going all over the place. About 15% of the cholesterol in our bodies comes from our diet, and the rest is made by our liver. So if you hear someone has high cholesterol and they're trying to decrease it with their diet, that's a challenge, because most of it is just getting made by our liver due to genetics. Let's look at a picture here. This is one of my favorites. Okay, so let's look at the structure of cholesterol first. So when I've taught this in the past, I've always told my students, if the structure looks like a chain link fence, it's related to cholesterol, okay? So no other thing that we have seen in any of these videos has this fused ring structure. There's nothing else like it. I think it looks like a chain link fence. I've had students tell me it looks like a honeycomb. Remember this general kind of shape. Be able to recognize it as cholesterol. You don't need to draw it. That's not necessary. But if you see something that looks like this, know that it's cholesterol or something closely related to cholesterol. Okay, so this is where it's cool. Here is estrogen, and here is testosterone. These are the two hormones that give us our secondary sex characteristics as human men and women. Do you see how similar their structures are? Let's look for differences. Okay, so here we have a hydroxyl group, and here we just have an oxygen. That changes the chemistry of this first ring just a little bit. Here we have 3 double bonds, and here we only have 1 double bond. We have a methyl group projecting between these 2 rings, and the methyl group is lacking here. And then everything else is the same. So those 3 seemingly minor differences account for huge differences in how our bodies function and how human and other mammals reproduce. It's really incredible to think how important simple chemistry changes are. So going back to cholesterol, we talk sometimes about good and bad cholesterol, and when we do that, we're talking about a ratio of lipid, the cholesterol, to certain proteins. And in one ratio, high density lipoproteins, we may have some prevention of cardiovascular disease. And if the ratio is flipped and we have more lipid and less of the specific protein we're talking about there, that can contribute to cardiovascular disease. So that's just a little bit about something you may hear in just everyday conversation: good cholesterol, bad cholesterol.